In this video tutorial, I am going to discuss about digestion of proteins. Protein digestion starts in the stomach. Remember, protein digestion does not start in the mouth because in the saliva there is no any enzyme which can digest protein. So, protein digestion starts in the stomach. In the stomach, there are gastric parietal cells and chief cells. These parietal cells of the stomach, they secrete hydrochloric acid. Hydrochloric acid is a strong acid, and when it mixes up with the foodstuff, it provides a pH of approximately 2. So, it creates an acidic environment. This acidic environment has certain advantages. The first thing is the antiseptic advantage. That means whatever fungi or bacteria are there in the foodstuff, they all get killed, so we are protected, right? The second thing, that strong acid, they can denature protein. So, denature proteins. Such denatured protein, it is easy to digest. Why denatured protein is easy to digest? Because denatured protein has a more surface area, so more of the peptide bonds will be accessible to the digestive enzyme, right? The second type of cell which are important for the digestion of protein is the chief cells of the stomach. These cells, they secrete pepsinogen. Pepsinogen is the enzyme which can break down internal peptide bond. But pepsinogen is not active form of enzyme, rather it is inactive form of enzyme. And as we will see further ahead in the video, that most of the enzyme which are related with the digestion of protein, they are almost all they are secreted in the inactive form. And this has advantage. What is that advantage? See, if pepsinogen is active inside this cell, what will happen? It can digest all the protein inside of this chief cell, and so ultimately this cell can be digested. So, it has potency to digest our own tissue, right? But thank God we have inactive form of enzyme, so our cells are undamaged. Once it gets secreted into the stomach cavity or intestinal cavity, then only it gets activated. So, our tissue is not digested, rather whatever we take up in the food, that can only be digested. So, pepsinogen, as it is inactive form, and such inactive enzyme, they are collectively known as zymogens. So, pepsinogen is one of the example of zymogens, right? So, here now once pepsinogen is inside the gastric cavity, it needs to get activated to its active form, that is pepsin. So, how pepsinogen is activated to pepsin? Actually, pepsinogen is a larger peptide chain. It has N-terminal and C-terminal. What happens? In the pepsin, this N-terminal 44 amino acids, they need to get removed. So, this N-terminal 44 amino acid sequence, it needs to get removed. So, initially hydrochloric acid, it carries out this function. It removes this fragment of 44 amino acid long from this N-terminal. Right. So, remaining polypeptide, that is known as pepsin, and it is fully active form, right. The problem with the hydrochloric acid is that it is a slow acting. That means it cannot rapidly convert pepsinogen to pepsin. So, what happens? Initially hydrochloric acid acts on the pepsinogen, and few molecules of pepsinogen gets converted to pepsin. Once we have few molecules of pepsin, this pepsin again acts on its own molecule, that is pepsinogen, and it will rapidly remove this N-terminal fragment. So, this process is the rapid, right. So, initially HCl will activate some of the pepsin, and then pepsin rapidly removes all of this pepsinogen's N-terminal fragment. So, within no time all the pepsinogen is converted to its fully active form, that is pepsin. Now, pepsin has a capacity to break down peptide bonds, but it has certain characteristics. The first thing is that this pepsin is active, fully active in the acidic environment. Its pH optimum is between 2 to 3, and we know that this pH is provided by hydrochloric acid. So, here where we had listed the functions of HCl in the stomach mucosa, we can add third function, that activation of pepsin, as well as it provides this optimum pH for the activity of this pepsin. So it also provides optimum pH, right? The second characteristic of pepsin is that it is endopeptidase. What is the meaning of endopeptidase? So to understand this, suppose this is the polypeptide chain where this circle represents amino acid and horizontal line represents peptide bond. Suppose this is the N terminal and this is the C terminal. Here all these bonds except for this one and this one, all these red marked bonds, they are the endo or internal peptide bond, and on this bond only endopeptidase acts, right? Whereas this terminal bond, this is the N terminal peptide bond and this is the C terminal peptide bond. At these places, whatever enzyme act, we call them as a exopeptidase. And as it is evident from this figure, it is clear that exopeptidase can be of two types. It can be N terminal, it can act on N terminal, or it can act on C terminal. If it acts on N terminal, we call it as a aminopeptidase. And if it acts on C terminal, we can call it as a carboxypeptidase, right? And whatever enzyme which act on this internal peptide bond, we call them as a endopeptidases. So this terminology must be clear: endopeptidase, exopeptidase, aminopeptidase and carboxypeptidase. Remember, aminopeptidase and carboxypeptidase, they are type of this exopeptidase, right? So once the idea of endopeptidase is clear to us, we can move to the next characteristic of the pepsin. Now pepsin, it not just randomly cuts down any internal peptide bond, but it has some specificity, that if certain conditions are met, then only it breaks down this internal peptide bond. What is that condition? First, it identifies certain amino acid only, right? So these certain amino acids are mostly aromatic amino acid like phenylalanine, tyrosine and tryptophan, right? In addition to this aromatic amino acid, it also identifies methionine. So once it had identified any of the four molecule in this polypeptide chain, let's say this is the specific amino acid for the pepsin. It can be any one of these four amino acid, right? Now with this amino acid, there are two peptide bonds, one on the N terminal side, second one on the C terminal side. So which peptide bond will be broken down by the pepsin? So it breaks down the peptide bond which is on the carboxyl side of this particular amino acid. So this peptide bond will be broken, whereas this will not be broken, right? So this is the specificity of the pepsin. So now pepsin is fully active and we understood its specificity and other characteristic. We can easily predict that this pepsin will act on the ingested proteins which we have taken in the food, and of course it is going to be a large molecule of protein. Suppose this is the protein molecule which we had ingested. So pepsin will act on this. It will break down this peptide bond and so break down of this polypeptide chain, right, with this specificity. And what we get? We get fragments of polypeptide. We call this as a partially digested protein. Partially digested proteins, right? So this is the maximum level of digestion we can achieve inside the gastric cavity. Now there is this phenomena that I described. It occurs in the adult. In case of infants, there is one more mechanism by which protein can be digested. We know that infants are mostly on the mother's milk or other milk. This milk contains protein. Main protein of the milk is casein. So what happens in the infant? Gastric mucosa secrete one more enzyme, that is renin. This renin is secreted only by the infantile stomach, not by the adult stomach. And remember, this renin is not same as this renin. You can see the subtle difference in the spelling of this both renins, right? This renin is important in the regulation of the blood pressure and it is secreted from the kidney. This renin has nothing to do with this renin. This renin is the enzyme which is secreted by the infantile stomach. So, what this renin enzyme will do on this casein protein? This renin enzyme, it carries out denaturation of this casein, and along with denaturation, it also makes this casein to bind with the calcium. So, ultimate thing which we get after action of renin is the paracaseinate. Paracaseinate. So, paracaseinate is nothing but the denatured casein along with its complex with the calcium. Now, this paracaseinate is, as it is denatured, it is easy to digest. So, this pepsin can act now on this denatured protein very easily. So, easy to digest by pepsin. And yes, this phenomena, in this what happens, this casein is a milk protein, but when it gets denatured in the form of paracaseinate, its appearance looks like a curd. Okay, this casein's appearance is like a milk, whereas this paracaseinate, its appearance is like a curd. So, we can call this process as a curdling of milk, right. So, this is how the digestion of protein occurs in the stomach. Now, let us look at that how the further digestion of protein occurs. So, once digestion is complete in the stomach, all the gastric content along with the acid, it will reach to the next part of intestine, that is duodenum. In the duodenum, what happens? The content from the stomach is received, and this content is obviously it is going to be acidic because of hydrochloric acid. We call such content as a acidic chyme. This acidic chyme, as soon as it arrives in the duodenum, it leads to stimulation of two hormones. Two hormones are getting released. One is cholecystokinin and second one is the secretin. Cholecystokinin, its older name or formerly it is known as pancreozymin. Pancreozymin. Let us first look at the action of secretin. What this secretin will do? Secretin stimulates pancreas, and it stimulates pancreas for the secretion of bicarbonate ions. This bicarbonate is the alkali. So, what happens? This bicarbonate, it neutralizes this acidic chyme, and so because of this bicarbonate, pH of this acidic chyme will now reach to the 8. So, it becomes alkaline. This alkaline pH has advantage in the duodenum. What is that advantage? Advantage is that now onwards, whatever enzyme which act on the protein, they will be active at this alkaline pH, right. So, this will provide a favorable environment for the further digestion of protein. On the other side, this cholecystokinin, what it will do? This hormone, it stimulates again pancreas, right. Here also pancreas was stimulated, but in case of cholecystokinin, pancreas is stimulated for the secretion of pancreatic juice. This pancreatic juice is nothing but the collection or mixture of pancreatic enzyme responsible for protein digestion. So, in pancreatic juice, we have mainly two types of peptidases are there. As we had seen earlier, we have endopeptidases and exopeptidases. So, there is a endopeptidases and exopeptidases in the pancreatic juice. What are different endopeptidases in the pancreatic juice? We have mainly three types of endopeptidases. One is trypsinogen, second one is chymotrypsinogen, and third one is the proelastase. These exopeptidases, we know that exopeptidases can be of two types. One is carboxypeptidases that we had seen earlier, that it removes or it breaks down the peptide bond on the carboxy terminal, and second one, its counterpart, is the aminopeptidases. So, carboxypeptidases which are secreted or which are there in the pancreatic juice are the procarboxypeptidase A and B. Procarboxypeptidase A and B. Remember, there are no any aminopeptidases in the pancreatic juice. Now, as you can see, there is a suffix of jan or prefix of pro are there. What does it indicate? It indicates that these all enzyme in the pancreatic juice, they are not active, they are actually inactive. And as earlier we had discussed, this inactive form of enzyme, digestive enzyme, they are collectively called as a zymogens. Right? So, all of these are zymogens. Now, this all needs to get activated so that they can act on the protein molecule, right? So, for the activation of this, what will happen? Duodenal mucosal cells, they secrete one more enzyme, that is enterokinase, which is also called as a enteropeptidase. What this enterokinase will do? Enterokinase, it acts on this trypsinogen. And from this trypsinogen, one hexapeptide is released. Hexapeptide is removed from the trypsinogen, and then trypsinogen gets converted to trypsin. This trypsin is the active form of enzyme. Now, it can break down various peptide bond. Then this trypsin, it will carry out activation of this chymotrypsin, proelastase and procarboxypeptidase A and B. How this activation takes place? So, this trypsin, it activates chymotrypsinogen to chymotrypsin. Proelastase to elastase, and carboxypeptidase A and B to carboxypeptidase A and B. Sorry, here it is a procarboxypeptidase. Okay, so procarboxypeptidase A and B to carboxypeptidase A and B. All of these three are activated by the common enzyme, that is trypsin. Now, this trypsin, chymotrypsin and elastase, all these three are endopeptidases, whereas carboxypeptidase A and B, they are the exopeptidases. So, what will happen? Now, what is the specificity of all of these four enzymes? See, trypsin, what it will do? It will identify basic amino acids. For example, lysine, arginine, they are identified by the trypsin, and then it breaks down the peptide bond which is on the carboxylic side. What about chymotrypsin? Chymotrypsin identifies aromatic amino acid. And again, here also carboxy side peptide bond is broken down. What about elastase? Elastase identifies small amino acids, specifically having aliphatic side chain, right? So, it identifies alanine, valine and leucine, and these amino acids, carboxyl side peptide bond is broken down. What about carboxypeptidase A and B? Remember, carboxypeptidase A and B, they are the type of exopeptidase which occurs on the carboxyl side, right? So, suppose this is the polypeptide chain, this is N-terminal and this is C-terminal. So, carboxypeptidase, they specifically break down this terminal peptide bond located on this carboxyl side. This carboxypeptidase A, it identifies aromatic amino acid. So, suppose if this amino acid is aromatic, then carboxypeptidase A will act, and carboxypeptidase B, it will act on basic amino acid. basic amino acid, right. So, if this amino acid is basic, then carboxypeptidase B will act. Now, as you can see, it is removing carboxy terminal amino acid, but the if question is tested that what peptide bond is broken down, you need to say that this specific amino acids, this side of peptide bond, that is N-terminal side peptide bond, right. So, it acts on this amino acid on the N-terminal side, but the amino acid which is removed is of carboxy terminal side, right. So, what will happen that on this partially digested protein, that is the ultimate digestion in the stomach, that will be further acted upon by all of these 4 enzymes with their specificity, and ultimately what we will get, because of this hexapeptidase we will get most of the free amino acids, and because of this endopeptidase activity we get certain oligopeptides. They are the shorter chain of amino acids, and it has having 2 to 6 amino acid residues. So, at the end of all this pancreatic juice enzyme action, we get this as the ultimate product of protein digestion. Now, this protein, this partially digested protein, they will reach to the further distal part of the small intestine, and this small intestine mucosa also, they secrete various peptidases or various enzymes which can break down peptide bond. So, what are these various enzymes? There may be various peptidases, then there are various aminopeptidases, then there is a dipeptidase and tripeptidase. See, various peptidases, these are the endopeptides which can further break down the product. Aminopeptidase, we know that we had already discussed this. See, meaning of aminopeptidase. Aminopeptidases are those peptidases which act on the terminal peptide bond on the amino side. So, this act as a exopeptidase and removes amino acid from the N terminal. These various peptidases, they are all endopeptidases. This dipeptidases, it acts on the dipeptide. Dipeptide has 2 amino acid and a single peptide bond. So, they act on this dipeptide. Tripeptidases, they act on tripeptide. Tripeptide had 3 amino acid and 2 peptide bond. So, tripeptidases act on either on this bond or on this bond. So, digestion of tripeptidases, action of tripeptidases, it will give free amino acid and dipeptide, which will be then acted upon by the dipeptidase, right. But in the small intestine, the efficiency of all these enzymes are not 100%. So, what we will get as the ultimate product of this digestion is free amino acids. Most of the protein will be now in the free amino acids, and some of the protein will be still in the di or tripeptide stage. So, this is the maximum digestion human can achieve by the digestion of protein. And remember, 5% of the ingested protein remains undigested. So, 5% of the proteins are undigested. So, this completes our digestion of protein. In the next video, I will discuss about the absorption of this finally digested product of protein digestion.